So organ shape is really important for all organisms. And if you think about something like Darwin's finches, and the shape of the bill is incredibly important for how those different species live. If you have a big bill, you can eat big seeds and crack them open. If you have a long, thin bill, you can get into wood and find insects inside the wood, like the woodpecker. So all organisms, we really want to know does shape evolve and what controls how different organs evolve into different shapes. So columbines are very much like Darwin's finches that we were just talking about. They've evolved into a lot of species very, very quickly, and they've adapted by changing the shapes of their flowers to different kinds of pollinators. So they've evolved the shape of these organs, particularly the petals, to match the tongue length of different pollinators and allow the plants to reproduce. So they're a really nice system to study how rapid evolution works and how adaptation to lots of different environments occurs. Well, what's really interesting about, again, about Aquilegia is these petals. I'll pull one off here. This is a single petal, and the petals have a tube. This is just a tube that goes down the petal, and at the very tip, there is a nectary that makes nectar and fills the tube up with nectar. And then pollinators have to come along and stick their tongue down that tube and to get the nectar, which is what they feed on. And when they do, they touch the reproductive parts of the flowers and move pollen to the stigma, and that allows seeds to be formed and the plants to reproduce. So what's critical in these plants is how long does the spur become? Because if you have a very short spur and a long-tongued pollinator, the body of the pollinator would be way out here and wouldn't touch the reproductive parts of the plant, and the plant wouldn't reproduce. The pollinator would get a reward, like get food, but the plant wouldn't get to make seeds. So the plant has to make this spur long enough to force the pollinator to do its pollination. And so different species of Aquilegia have evolved from very short spur species that are pollinated by bees that have fairly short tongues. And from those we've shown before that a number of times they've evolved into hummingbird pollinated plants that have a slightly longer spurs. Hummingbirds have a longer bill and a little bit longer tongue, and that fits them. And then from hummingbird pollinated plants, we've been able to show that they evolve into hawk moth pollinated plants. And hawk moths are these great big moths with really long tongues. They curl up and they can uncurl them and stick down much further down a longer tube. And there are hawk moths that have tongues this long, and there are hawk moths that have tongues this long. And so different species of Aquilegia have evolved their spur even longer to fit those tongues as well. So we have this really nice system where we understand the ecology of why the plants would want to make a longer and longer spur to match different pollinators. And it's a fairly simple organ to try to understand its shape. And what we had known or what people had suggested for the last 60 years is that to get a longer spur, what you do is you just add more cells. And so a really long spur would have many more cells, and a shorter spur just wouldn't have very many cells. That was suggested, but no one had really looked at it for 60 years. It was just assumed that that had happened. And so Josh and Sharon, the two senior authors on the paper, the graduate students, set out to measure this. And they went in to count the number of cells and look at what cells, the shape of the cells looked like. And what they found is that it's not adding more cells. A spur that's this long has just as many cells as a spur this long. And what's the difference is the shape of the cells. The cells get progressively longer and thinner, so they grow mostly in one direction rather than in width. and it's not adding new cells. And that's quite unusual for the control of shape of any organ in plants that has ever been studied before. So this is kind of a new mechanism that people hadn't recognized before for how organ shape can evolve in plants. And we think that this might be a common way for other organs that people haven't studied very much. Often in other species, the stamens here get longer in some species, or the styles here, this part of the plant that receives the pollen gets longer, depending on the evolutionary circumstances. And we think that maybe people should be looking at that as well, because it just hasn't been considered previously. So one of the things that we did in this study was to measure exactly that. And we would take buds of flowers, very, very small buds, when you just started to see the spur. And every day we would measure how long the spurs got. And what was really interesting is that all the species, and we studied little short spurred species and the really long spurred species, spurs grew at the same rate. And so the short spurred species, though, once it got to a certain length, then it stopped growing, the flower opened, the anthers released their pollen, and the stigmas were ready to receive pollen so that they could reproduce and make seeds. The other species just kept on growing, and the flowers had to kind of be in suspended animation. They had to wait before they opened and were receptive and ready to have sex until the spur got longer. And even the longest species, it took them almost a week longer to grow. They grew at the same rate at the beginning. The others stopped growing. They just kept on growing for another week. And then their spurs were so long, this long, and then the flowers opened and were ready to be receptive. So what's really different among these species is how long the flower buds are available to grow before they're reproductively active.